We will wait for another minute and then I will start the presentation. Okay, let's start. Someone please confirm the slides are changing. Hello, am I audible? Hello, hello.
Excuse me, ma'am.
Yes.
Ma'am, you are audible and the screen is visible too.
Okay. Okay, we'll start. So I welcome you all to week 3 session for the functional genomics problem solving session. So in the last discussion, we discussed about various techniques which are used for the which are used to study the mRNA. So I was left with one more topic, which was PCR. So PCR stands for the polymerase chain reaction, and it is used to amplify the DNA of known sequences. A PCR cycle simply involves the three steps, which is denaturation, annealing, and extinction. So in denaturation step, firstly, the double-stranded DNA, which is the usual form of the DNA in which it is present, it is denatured by the process of heating at approximately 94 degrees centigrade. This leads to the production of two strands, and after that, in one PCR cycle, what we do is we anneal. The next step is annealing, where we anneal the primers. These oligoprimers are bind to the target sequences which we want to amplify. And after that, the third step is the extension, where the DNA polymerase or the Tech polymerase, which we used for the PCR reaction, are used, and this helps in the extension of the targeted sequences. Thus, from one DNA template strand, which is double-stranded, we can obtain the two copies of the similar DNA. So, what are the properties of the primer or the oligomers which we are using at the annealing steps? So, what are the desired properties which should be of the primer? So, firstly, while designing a primer, we should always keep some points. There are some points which we should always keep in the mind. Firstly, as I have highlighted in this slide, in the first point, the aim for the GC content should be between 40 to 60%, and the 3 prime end of the primer should, if it is, it would be better if it can end as G or C, as it promotes the binding. As we know that the number of bonds between the G and C in the double-stranded DNA is 3, while as compared to the A and T, they are 2. So, it is important that when we are designing the primers, we should take care of how much the percentage of GC is there and how much percentage of AT composition is there. And it is always preferred that for the primer, the 3 prime end, if it is G or C, it would be better as it promotes the binding of the primer with our single stranded DNA. Uh, The second point is that it should not have too many repeated G or C, as it may lead to the primer dimer formation. As the G may form a bond with C, and it may leads to the dimerization or some hairpin loop like structure formation in the primer. Next point is that the good length of the PCR is usually 18 to 13 bases, and this length uh, affects the PCR products. The shorter the primers, the more efficiently they will bind or anneal to the target. But if we take residues, like suppose we take 8 to 10 residues or very less number of residues, then what will happen is it can have the unspecific binding also. So it is very important to keep in mind the length. It should be between 18 to 30 bases. And the next point is very important. That is, we should always try to make the melting temperature of the primers between 65 to 75 degrees centigrade and within 5 degree of each other. Within 5 degree of each other, I mean that for the forward primer, let's say it is 65 degree. Then for the reverse primer, it should be within the 5 degree of that forward primer. This is because if there is a very large difference between the annealing temperature or the melting temperature of the primers, then there will be a problem in annealing. Also, we know that the bases also impact the melting temperature, as we know that G and C results in the higher melting temperatures as compared to A and T. And if the TM, that is the melting temperature of our primer, is very low, we can increase a little bit of the GC content, or we can increase the length of the primer in order to get an appropriate melting temperature. Typically 3 to 4 new. In case if we are using the primers and then we will be using the PCR product in the cloning, and we want to use the restriction enzyme further, then it is preferred that 3 to 4 nucleotides should be added at the 5 prime end of the restriction enzyme site, and that in the primer, to allow for the efficient cutting. Also, as I mentioned in the first point itself, that we should try to avoid the secondary structure formation by balancing the distribution of the GC and AT rich domains. Next point is that we should avoid the runs of four or more of one basis. For example, like there should not be a consecutive similar types of bases. We should avoid that kind of situation. For example, we can see that the sequence A, C, C, C is not a good primer sequence. Similarly, AT, AT, which is the repetition of a similar kind of base, is a similar pair of bases is also not a very good primer property. The next point also I have previously explained that we should avoid the intra-primer homology, so that there should be no base pairing between the components of the primer itself, and also we should avoid the inter-primer homology. That is, the forward primer and reverse primer should not be complementary to each other, as if they will be complementary, they will bind itself with each other and they will lead to the formation of primer dimers. So there are various sites which are available where we can go and design our primer. One such is Primer Blast, and this has been described in the tutorial section of this NPTEL course. So you can go and check that video, that how to design a primer using a Primer Blast. So this window has many options. Like you can give the sequence, and you can define the range from where to start the primer and from where to start the reverse primer. And similarly, you can mention the melting temperature which you preferred, and there are many other properties which you can go through. And in case if you have any doubt, you can post it on the forum. Uh, After designing the primer, the next thing is we should analyze the properties of the primer. So we should analyze the properties which all the properties which I have mentioned in the previous slide. So there are certain tools like oligocalc which calculate the properties of the oligonucleotides, like it calculate the tm of the oligonucleotide, at what concentration the tm of the sorry at what like you we can calculate the gc content of the primer, we can calculate the length of the primer, we can calculate uh, the other whether it will lead to the primer dimer formation or not, we can check all these things. Similarly, there is oligo analyzer tool, where we can quickly see the GC content, melting temperatures, whether there is any secondary structure formation potential in the primer, or whether there is a property of dimerization in this primer. So we can use these tools to check whether the primer which we have selected for the amplification is appropriate or not. There is another tool which is also known as oligo calculator. So you can just mention the primer sequence here. You can mention, it will give you the length, melting temperature, and what is the OD in case if you will want to measure the concentration of the primer. So one of the important properties, which is the most important, is the melting temperature of the primer. So this is the basic calculation for the melting temperature of the primer, which you can also use these equations to manually calculate what is the melting temperature of your primer. So as we know that the melting temperature of the primer depends on the nucleotide composition. So what how many number of A and T are there and how many number of G and C are there. So based on that, if our sequence length is less than 14 nucleotide, we can use this equation where the melting temperature is equal to the number of nucleotides, number of adenine which are present plus number of thymine which are present multiplied by 2 pl plus the number of guanine and cytosine multiplied by 4. And if we are using the longer sequence, which is longer than the 13 nucleotide, we can use the equation which is mentioned here. So where X, Y, Z, W, X, Y, Z are the number of bases of ATGC in the sequence respectively. But there is an assumption behind this equation, and the assumption is that both the equations assume that the annealing occurs under the standard conditions where we are using the concentration of 50 nanomolar primer concentration, and the salts which are present, that is the sodium salt which is present, is in concentration of 50 millimolar and at the pH of 7. So the first question is very similar to what was asked in the assignment 2. So in a polymerase chain reaction, how many DNA duplexes can be obtained from 4 DNA duplex after 4 cycles? So if anyone would like to answer that. Hello, would anyone like to answer this question? Like in a polymerase chain reaction, how many DNA duplexes can be obtained from 4 DNA duplex after 4 cycles? Okay, so Bhagirath has answered 64. Anyone else who would like to attend this question? Hmm. If anyone would like to explain how did they get the 64? How did they come up to the number 64? Okay, so most of you have given the answer option D, which is 64, and which is also the correct answer. So it is very easy, as we can see in this DNA, this PCR reaction, where a single DNA duplex molecules gives the two similar DNA molecules at the after the extension. So in this question, we can simply say that there are 4 DNA duplexes. So right, so Ricky has answered that the number of the number of DNA molecules which will be obtained after one cycle will be 2 into 10 to the power, sorry, after n number of cycles will be 2 into 10 to the power n. And since there are 4 DNA duplexes, this will be multiplied by the 4. And n will be also equal to 4 as there are 4 cycles which are taking place. So the next question is, in a polymerase chain reaction, which is a widely used technique to amplify the target DNA, what would be the outcome of the PCR if the annealing temperature is higher than the extinction temperature? So, anyone who would like to answer this question? So, whether primers will not be able to bind to the template, whether the single-stranded amplification will take place, PCR tubes will melt, or the amplicon will be smaller than the expected size. So, you can attend this in the chat box also. What would happen if the PCR annealing temperature is higher than the extinction temperature? Primer annealing temperature is higher than the extinction temperature in the PCR. Okay, if no one wants to answer, I'll answer this question. So what will happen is that the primers will not be able to bind to the template. As we know that in a PCR, the last step is the extension. So there is a cycle in this PCR cycle, the denaturation usually held at 94 degrees centigrade. Then annealing is depends upon the primer composition, but it's usually preferred between 60 to 70, as I said. And then extension is carried out at 72 degrees centigrade. So after the annealing, that is the after the binding of the primer to these template DNA strands, the tech polymerase comes and extend the DNA according to the based on the complementary sequences. So what will happen is if we increase the annealing temperature, then the extension temperature, then the primers will not bind and the extension will not takes place further, and therefore we will not be able to get our PCR product. So if the temperature will be high, the primers will not be able to bind to the template, and therefore will our PCR reaction will fail. One of the important type of PCR is the RT-PCR, which is the reverse transcriptase PCR, and it, as the name suggests, here we for what is the purpose of this RT-PCR is to convert the mRNA to cDNA, and then we can amplify the DNA to have the understanding or to use it for the various genomic information. So this messenger RNA, which is polyadenylated, which have a polyadenylated tail, undergoes the RT-PCR reaction in the following manner. Firstly, uh, either it can have a poly-DT primer, either we can design a poly-DT primer, or we can design some random primers which will bind randomly to this mRNA sequence, or we can design a specific primer which is specific for a certain sequence present in the mRNA. Then the enzyme which will be used here is the reverse transcriptase enzyme. As we know that the function of the reverse transcriptase is to uh, uh, reverse transcriptase enzyme is to convert the to help converting the mRNA into the DNA. That is helping in the DNA synthesis, cDNA synthesis basically. So when we'll provide the reverse transcriptase, the appropriate buffer and DNTPs will be provided. It will lead to the formation of cDNA mRNA hybrid. So this cDNA and mRNA hybrid, then we use the enzyme which is also known as RNase H. This RNase H enzyme actually helps in the denaturation of this hybrid, which is the cDNA and mRNA hybrid, and it cleaves the gaps in the mRNA strand. Then DNA polymerase 1 comes and it leads to the formation, it completes the strand of the DNA complementary to the earlier strand which was formed in the first step, and therefore leads to the formation of a double-stranded complementary DNA. So this is the procedure of RT-PCR. If anyone has any doubt regarding the enzymes which we are using or the procedure which we are following, is it clear to all? Okay. So based on your understanding of different approaches which are used to quantify specific nucleic acid targets, identify the options correctly depicting the name of the approach represented in the given image. So is it a representation of a polymerase chain reaction, or is it a representation of reverse transcription polymerase chain reaction, that is RT-PCR, or is it a description of microarray or northern blotting? So anyone who would like to answer? Okay, Smriti has answered option number B, which is reverse transcription. Okay, so yes, it is the representation of the reverse transcription polymerase chain reaction. The next question is, identify the enzyme that is not used for the construction of a cDNA library. So which one of the following enzyme is not used in the construction of a cDNA library? First option is RNA dependent DNA polymerase. Second option is RNase H. Third option is DNA dependent DNA polymerase. And fourth option is DNA dependent RNA polymerase. So anyone who would like to attend this question? The enzyme that is not required for the construction of a cDNA library. Okay, Divya has answered option D. Bhagirath also has answered option D. Anyone else? Anyone who would like to explain why option D for those who have answered? Divya and Bhagirath. Okay. So
In the,
huh,
in the construction of DNA with the help of RNA, we are using RNA dependent DNA polymerase and
okay.
removal of RNA single stranded RNA. RNase H enzyme is helpful.
Yes.
Normally, we are using DNA polymerase 1, 2, 3. It is DNA dependent DNA polymerase only.
Yes, yes.
Left over D only. That is the wrong answer.
So you think that DNA dependent RNA polymerase is the wrong answer.
Yes. Yes.
Okay. Anyone has any doubt or is it clear to everyone? Okay. So the next question is messenger RNA, that is the mRNA, can be quantified by which of the following functional genomic technique? Is it microarray or dot blot analysis, RT-PCR, and all of these? This is a very easy theoretical question. Which of the following techniques can be used to identify the mRNA or quantify the mRNA which is present in our sample? So Ricky has answered option D. So this is a easy theoretical question. I'll move forward. So the correct answer is option D. All the techniques like microarray, dot blot analysis, and RT-PCR can be used to quantify the mRNA which is present in our sample for the functional genomic techniques. So the next concept which was introduced in this week 3 module by Professor Ganesh was DNA sequencing. So DNA sequencing, firstly, the structure of DNA was Watson and Crick identified the double helical structure of DNA around 1950, and after that, the era of the sequencing started after the first sequencing by the Sanger. So Sanger started work on the DNA sequencing in around 1970, and in around 1976, first complete RNA genome of a bacteriophage was sequenced. And later, Sanger developed the dideoxy chain termination method. Then came the Maxam and Gilbert chemical degradation method that we will discuss later in this presentation. And after that, in the Human Genome Project, we have used multiple techniques for the sequencing. And now we have an era of next generation sequencing. And we also have third generation sequencing. Next generation sequencing is also known as the second generation sequencing, and the first generation sequencing involves the Sanger sequencing and the Maxam Gilbert sequencing. So this is just a brief classification of the different sequencing techniques in three generations, which is first generation, second generation, and third generation. So first generation involves the shotgun sequencing, Maxam Gilbert sequencing, and Sanger sequencing. Uh, In this, the sequencing is usually done by the synthesis, which involves the principle of which is based on the normal DNA replication. So, and it has a high accuracy, long read length, and relatively small amount of the data is generated. For example, the ABI capillary sequencers. In the second generation sequencing, which is also known as the next generation sequencing. It involves the massively parallel sequencing, and in this, the sequencing is also done by synthesis. Amplified templates are generated during sequencing, reducing the requirements for starting material. Accuracy is high and short read lengths are required. The read lengths are short. Example, MISEC, which is Illumina, Ion Torrent, Thermo Fisher Scientific. This also we will discuss later. And in the third generation, what we'll do is we are trying to study the single molecule, we are trying to perform the single molecular sequencing. And in this, the single template is obviously a single molecule. It has a low accuracy, but has a long read length. So example include the single molecule real-time SMRT sequencing and Minion, that is, which is the technology of Oxford Nanopore Technologies. So most of the DNA sequencing is based on a procedure based on a normal DNA replication, where we know that the if we have a 3' to 5' DNA template strand, our primer comes and bind at the 3' sequence, and after this the DNA polymerase comes and it leads to the addition of different nucleotides based on the complementarity of the template strand, and therefore the new DNA will be formed with the help of the primer and the addition of the nucleotides using the DNA polymerase enzyme in the 5' to 3' direction. So during the DNA replication, what happens is that there is a formation of a DNA phosphodiester bond. So in DNA, we all know that the sugar which is present is the deoxy sugar, and in which the 2' do not have a OH group. Instead, it has a normal hydrogen group. So but the 3' end of this the 3' has the OH position, OH group, and it is the position where the next nucleotide will come and bind. So when the next nucleotide come, firstly the bond breakage happens, leading to the release of the pyrophosphate, and this monophosphate and nucleotide base binds to the previously uh, added nucleotide base, thus leading to the formation of the complete DNA strand. And this bond, which is formed with the first 3' OH with the 5' phosphate group which is present, is the phosphodiester bond. So the principle on which the Sanger sequencing is based is that in Sanger sequencing, we do not have this OH group on the second prime. We do not have it utilizes 2' 3' dideoxyribose sugar, in which we do not have a OH group at this 3' end. Also, therefore, whenever this kind of nucleotide will be added up in the DNA sequences, it will further stop the elongation of the DNA molecule, as there will be no free OH group where the phosphodiester bond formation can take place. So this is the basic principle, and therefore this is how the dideoxyribonucleoside will block the elongation, as it do not have any 3' reactive hydroxyl group. So the Sanger sequencing method, which is also known as the chain termination method, as we are terminating the formation of the DNA chain here, uh, involves the following step. Firstly, we set up a normal DNA polymerization reaction, which consists of a template strand, a primer, and DNA polymerase, and different nucleotides. But beside all these normal nucleotides, we also have 2', 3' dideoxyribonucleoside triphosphate chain terminators, which are fluorescently labeled and are added in the reaction mixture. So we then incubate this reaction mixture and we try to see the synthesis of the chain termination. So whenever there will be a termination, there will be a fluorescent signal that is generated, and this fluorescent signal will be recorded. And we'll get a readout something like this, and from this we can see, like from this sequence what we have obtained here, we can see based on the fluorescence which we are obtaining, since all the four different dideoxynucleotides have been fluorescently labeled with different fluorophores. So based on the fluorescence which we are obtaining, we can detect what is the nucleotide which has terminated the chain. So the next is the Maxam Gilbert sequencing. Anyone has any doubt in this Sanger sequencing method? Hello? Okay. If there are no doubts, we can move to Maxam Gilbert method. So Maxam Gilbert sequencing is based on the chemical degradation, or we can say it is based on the use of the different chemicals that will that will stop the reaction, that will cleave the DNA at the different nucleotides. So firstly, the double-stranded DNA molecule is labeled at both the 5' and 3' at both the 5' ends using a phosphate molecule, and then we cut this labeled DNA strand with the restriction enzyme. The long DNA molecules are denatured, and after that, these single-stranded molecules are incubated in different reaction tubes, which consist of different compositions of the different chemicals that will, in the first reaction tube, the cleavage will occur after the presence, wherever the G will be present. In the second reaction tube, there will be a cleavage wherever there will be a or a G. Similarly, in the third reaction tube, the termination, sorry, not the termination, but the chemical degradation will occur wherever there will be a C or a T present in our template, and in the fourth tube only where there will be a C, it will get cleaved. So reaction proceeds long enough to produce an average of one break per strand, and the random breaks generated and labeled fragments representing the position in their representing their position in the sequence. So after cleaving and after providing the after incubating for the long enough time, we then run these uh, fragments which we have obtained in different test tubes in the gel. So we know that in the agarose gel electrophoresis, we will obtain the shorter fragments at a lower side of the gel, and we'll obtain the longer fragments near to the well where we are loading the gel. So this can be read out in the following way. For example, we see that the shortest fragment is formed in the tube 3. So in tube 3, what was there? It was C chemicals that will either cleave after C or will cleave after T. So how we will say whether it is a C or a T? So we will look at whether there is a bend in the lane of the tube 4 also or not. So since tube 4 do not has any bend in this position, therefore we can say that it is the T which has got cleaved here. Similarly, it will move forward. So we will see that we have obtained the bend in both the lanes, which is tube 1 and tube 2. So in tube 1, G was present, but in tube 2, A and sorry, okay. So in tube 2, that is in lane of the tube 2 also, the band is present. In tube 2, A and G are present. Sorry, the chemicals which will cleave after A or the G residues are present. So we see that since the band is present in both the lanes, it will be a G. And if it would have been present only in tube 2, then it would be a A. So this is the way how we read the sequence and in the Maxam Gilbert sequencing. So the next question is, if one of the in one of the popular sequencing method of DNA, which is the Sanger sequencing, selective incorporation of dideoxynucleotide triphosphate, that is DDNTP, is used during in vitro DNA replication because first option is DDNTPs emit fluorescence once they are incorporated into the strand of DNA. Second option is DDNTPs incorporates into the DNA sequence and prevents the DNA synthesis. Third option is DDNTPs can be purchased easily. They are very cost effective. And fourth option is all of these. So anyone who would like to attend? Okay. Divya has answered B. Anyone else who would like to attend? Okay. Ricky has answered A. So any one of you who would like to explain, like since both of you have answered, Divya and Ricky, and you have answered differently, like Divya has said option B and Ricky has said option A. Any one of you who would like to comment why you have chosen this option?
I think whenever the DDNP causes a disruption in the process of making the phosphodiester bonds between the nucleotides of one to another, so it is incorporated, and then only it can emit the fluorescence reaction once it gets when it causes any disruption to the strand, uh, and it stops the process of making the phosphodiester bond.
Oh, right. You have explained it correctly, but Divya, would you
Yeah,
like to explain why you have
yeah,
chosen option B? So
Ma'am,
basically, yeah,
DDNTPs prevent DNA synthesis
oh,
because they do not have free OH.
right. So, Ricky, you answered it correctly, but the principle for which we are using DDNTP is not because it emits fluorescence. It is emitting fluorescence because we have attached a fluorophore to it. You are right, but the principle because of which we are using DDNTP in this method is that we want to prevent the DNA synthesis, right? Is it clear?
yeah, got it.
Okay. The next question is, which one of the following statements correctly identifies the difference between the Maxam Gilbert method and Sanger sequencing method? So, Maxam Gilbert method is a chemical based sequencing method, while Sanger method is based on enzymes. Sanger method can be used to sequence both genomic DNA and the cDNA, while Maxam Gilbert method cannot be used for the complementary DNA. Option C, Maxam Gilbert method can use RNA as the template, while Sanger method can only use DNA as a template. Option D is none of these. Anyone who would like to answer? Okay. So people have answered option A. So the correct answer is option A. Maxam Gilbert method is a chemical based sequencing method as we see, and Sanger method is based on the use of various enzymes and DDNTPs. The next method is shotgun sequencing. In this shotgun sequencing, firstly, the original genome is broken up into several fragments and many small fragments are obtained. After this, we incorporate these small fragments into our clone and we clone these fragments and sequence these small fragments. After sequencing these fragments, what we do is we try to align these fragments by finding the repeated sequences, overlapping sequences, sorry. So wherever there will be a overlapping, it is suggested that the overlapping sequences help in defining the complete sequence of the DNA fragment. So there are various contigs which are obtained and there is a gap between the different contigs. So the contigs, then there will be some other contig that will fill up this gap, and based on this we define the complete sequence of our DNA or the RNA which is present, for which we are doing the sequencing. So after this there is an era of next generation sequencing, and the procedure which is also known as the second generation sequencing. The procedure for the next generation sequencing or the NGS is this is the basic scheme which is of representing the next generation sequencing. So in this, firstly, we isolate the genomic DNA. So genomic DNA is isolated and it is fragmented randomly. So random fragmentation takes place. After this, we then add the adapters to this random fragments and using the library adapter ligation. So this adapters are ligated with the DNA. These adapters, after this, we amplify these. ligated molecules. So this leads to the amplified DNA clusters, and after this we sequence these DNA cluster and we got the data for the analysis. So analysis also involves can be of three types, whether we can analyze the variants. So if there are some variants, variant analysis can takes place. If we are or we can map it with some reference genome, and then we can sequence the DNA which we have isolated, or it can be a de novo assembly, which is the new assembly which do not have any reference genome. So these are the five basic steps. Firstly, the genomic DNA isolation. Second is the next generation sequencing library construction, which involves two steps, that is the gDNA fragmentation and then modifying these strands by adding the adapters. The third is partitioning the library fragments into the separate locations on a solid surface and create the clusters of identical copies. Fourth is the sequencing of each cluster, and fifth is to analyze the data. So there are various techniques which are used for the amplification of the template, as this is one of the very important step where we'll get the amplified DNA cluster, only after which the sequencing can takes place. So there are various strategies for the template amplification. The most common are the emulsion PCR, or second is solid phase bridge amplification. So there are different platforms which are available for the sequencing methods. So there are different platforms like like Illumina or 454 Roche, or solid platforms, or there are certain platforms by QIAGENE like GeneBreeder, or ion-torrent by the Thermo Fisher. So these different platforms utilizes the different template amplification strategies. For example, emulsion PCR is utilized by 454 Roche, Solid, which is a Thermo Fisher platform, and GeneReader, which is a QIAGENE platform, and ion-torrent, which is again a Thermo Fisher platform. Okay, somebody wants to join. I don't know what happened. Am I audible? Okay. Sorry for the disturbance. Someone wants to join the meeting. Okay. So there are various different amplification strategies which are used. So the first one is the emulsion PCR. Emulsion PCR is used by the 454 Roche and solid platforms, and the solid phase bridge amplification PCR is used by the Illumina. So what happens is, in emulsion PCR, firstly the fragmented DNA template, which is the previous step, which involves the DNA fragmentation, are ligated to the adapter sequences and are captured in an aqueous droplet, which is known as the missile. And along with a bead that have the complementary adapters to which our DNA fragments can bind. So the emulsion, in which the micelle droplets are loaded with the primer, template, DNTPs, and polymerase. After this, uh, when our fragmented DNA, which is attached with the adapter, is comes and binds to this on the surface of this emulsified under this emulsion, it undergoes amplification. So templates hybridize to bead-bound primers and are amplified after amplification. The complement strands then dissociates, leaving the bead-bound single-stranded DNA templates. And this is the final product, which contains the single-bound DNA templates. And 100 to 200 million beads with thousands of bound templates will be obtained, which will further be sequenced using various sequencing techniques that we will discuss later. The second is the solid phase bridge amplification, in which the uh, uh, binding takes place at a solid surface, and the firstly free templates hybridized with the side bound adapters, and after this the amplification occurs, and amplification occur in a bridge like manner. So the distal ends of the hybridized templates interact with the nearby primers where amplification can takes place, and since they are interacting with the nearby primers, it will lead to the formation of a bridge like structure. So it And after the amplification, again, the cluster formation takes place. So there will be a generation of the clusters. So after several rounds of amplification, 100 to 200 millions of clonal clusters are formed. Clonal clusters matlab which are exactly similar. And the third is the solid phase template walking, in which the free DNA templates hybridize to the bound primers, which are already bound to the solid surface, and the second strand is amplified. The step is the primer walking. In this, the double-stranded DNA is partially denatured, allowing the free end to hybridize to a nearby primer. Therefore, in this, the bridge-like structure is not formed, as we see in this solid phase bridge amplification. The next is the in-solution DNA nanoball generation, which is used in this third generation sequencing. So in this, the adapter ligation happens, and one set of the adapter is ligated to the either end of the DNA template, followed by the template circularization. So in this, the template circularization takes place. After this, we cleave it. So the circular DNA templates are cleaved downstream of the adapter sequences, and then we use the iterative ligation, and therefore generating the large number of the copies for same clones. So this is the amplification step, where we can either amplify using the emulsion PCR or the solid phase bridge amplification PCR. These two are the most common. After this, we need to sequence our DNA. So the first is the sequencing by ligation. Sequencing by ligation technique is used by the solid platform, which is a Thermo Fisher platform. So in this, what happen is, following cluster generations or bead deposition onto a slide, fragments are sequenced by the ligation, in which a fluorophore labeled two probes are added, and this is composed of the nucleotides in the first and the second position. As we can see that the here this highlighted by this blue color, which is A and T, they are the known bases. Probe with two known bases followed by the degenerate or universal base hybridized to a template. Then ligase immobilizes the complex and the slide is imaged. After this, what we do is, after we analyze these two nucleotides, then we cleave it again. The fluorophore is cleaved from the probe along with the several bases, revealing a 5' phosphate, where another set of the two nucleotides will come and bind. So in this way, probe extinction will take place. So 10 rounds of hybridization, ligation, imaging, and cleavage identified 2 out of every 5 bases. Did you get this point? How 2 out of every 5 bases? So after this, we reset the reaction by moving 2 steps further. So after a round of probe extension, all the probes and anchors are removed, and the cycle begins again with an offset anchor, which is n plus 2 offset. So this way it moves every time 2 nucleotides ahead. And in this way, we get the sequence of our DNA molecule. The next is the sequencing by synthesis, which is used by the Illumina platform. So sequencing by synthesis involves the 3 basic steps. Firstly, it involves the nucleotide addition. Second is the imaging. And after this, it also involves the cleavage, where we'll cleave the fluorophore. And after the cleavage of the fluorophore, the 3' OH will be available for the binding of further nucleotides. So first step is the nucleotide addition, where the fluorophore labeled terminally blocked nucleotides hybridize to complementary base, and each cluster on a slide can incorporate into a different base. After this, slides are imaged with either 2 or the 4 laser channels, and each cluster emits a color corresponding to the base which is incorporated during this cycle. When we image this slide, after that, when we have to cleave the fluorophore, and fluorophores are cleaved and washed from the flow cells, and the 3' OH remains the free, is regenerated, and therefore it will lead to the new cycle beginning with the addition of the new nucleotide. And after the addition of new nucleotide, we follow the same step again. The next kind of sequencing is the pyrosequencing. So pyrosequencing involves the generation of the light which is detected, and in this case what happened is, in pyrosequencing, Till now, if anyone has any doubt. Okay. So, in pyrosequencing, it is based on the principle of the generation of the light, and this light is then detected by the CCD cameras. So, it is based on the principle that when a base is incorporated, it leads to the release of an inorganic pyrophosphate, and this triggers an enzyme cascade. So we know that addition of the base during the DNA synthesis leads to the release of the pyrophosphate, as I showed previously. And after this pyrophosphate actually converts what? It leads to the activation of the enzyme luciferase, and this luciferase leads to the formation of the protein, which is luciferin, and this is a light emitting protein. So based on the emission of the light, we can record the nucleotide which is getting attached. So it is a single nucleotide addition. Only one DNTP species is present during the each cycle. So we'll add, like in cycle one, we'll add only one kind of DNTP, and we'll see whether the it is emitting light or not. After this, similarly in the cycle two, we'll add the other kind of DNTP. So in this way, in the different cycles, different DNTPs are incorporated, and during the process of the extension, when there will be the binding of the next nucleotide, there will be the release of the pyrophosphate group, and this pyrophosphate group will therefore leads to the this energy dependent mechanism, enzyme mechanism, which will lead to the formation of a luciferin and ultimately resulting in the production of the light. The next is the ion-semiconductor sequencing, and it is based on the principle of the generation of the H plus, which is generated, and due to the production of the H plus, there will be some changes in the which can be detected by the detector. So, as a base is incorporated, a single H plus ion is released, which is detected by CMOS ISFET sensor. So, after the detection of this H plus ion, which is released as a result of the incorporation of the bases, this will give us a signal, and depending on what nucleotide we have added, only one DNTP species we are adding during the E cycle again, as we did in this pyrosequencing method. And based on this, if the H plus ion is getting released, we can say that it is that particular nucleotide which is going in binding, and therefore leading to the ultimate sequence of the DNA. So this is the comparison of the important next generation sequencing platforms. Uh, so there are various platforms which are present, like as I mentioned, ROS 454, Illumina, Solex, ABI Solid, iTorrent. So these are the different properties on which they are based, and they have certain advantages and disadvantages over each other. For example, if we are using ROS 454, which utilizes the emulsification technique for the amplification of the target DNA, and pyrosequencing for as a sequencing method. The advantages are that it has a longer read length and the small data files are generated. But the concerns are that we have a very less data, and therefore it is difficult. Sometimes it becomes difficult to say whether we are getting it correctly or not. So every next generation platform has its own advantages and disadvantages. So you can go through this slide when you'll read it again. And if you have any doubt, you can ask in the next session. Now, what are the applications of this next generation sequencing? This next generation sequencing is not just limited to the sequencing of our genomic DNA, but it is also used for the sequencing of the total RNA content which is present, which is known as the small RNA seq, or we can sequence the mRNA which is present, which is also known as the RNA seq. We can perform by performing the by checking the interaction of the DNA with the different proteins. That is by using the technique ChIP-seq. We can check whether what different proteins are interacting with different DNA fragments. So ChIP here, I would like to say what is ChIP. So ChIP is the chromatin immunoprecipitation technique, and this actually helps in immunoprecipitating, as the name suggests. Using the antibody, it helps in precipitating the complex of the DNA with the proteins. For example, if you want to study the histone proteins, So we know we can generate the antibodies against those histone proteins, and we can pull out a DNA sample using those antibodies. So the DNA sample which will be pulled out when we are using this antibodies against the histone proteins will help us in identifying the sequence where these histone proteins are getting are interacting with the DNA. So this is what the chromatin immunoprecipitation technique is. Also, we can go for the single molecule sequencing, as we know that the third generation sequencing involves the single molecule sequencing, or we can go for the amplicon sequencing. So these are the various applications and their interconnections which I have shown here for the next generation sequencing. Also, next generation sequencing has its application in the field of epigenetics, in the field of evolutionary research. If you want to, if you want to study, if you want to produce some transgenic animal, it is important to know the genomic DNA. Like it is important to have its genomic information. So it is important in the field of generating some transgenics. It is important for food safety and nutrition. It is important for improvement of the livestock, beef cattle selection, and if we want to study the genomic variability, we want to study single nucleotide polymorphism or the copy number variations, we can use NGS. Or if we want to do any kind of DNA research or we want to go for the metagenomics, NGS is a offers wide applications. So the next question is, the next generation sequencing refers to the contemporary methods of DNA sequences that revolutionize the genomic research. Identify which one of the following option is true with regard to the genomic DNA, that is the gDNA being analyzed in the next generation sequencing technology. So option A is the gDNA analyzed and fragmented in the process is heterogeneous in nature. The fragmented gDNA is then multiplied in various number of copies of a region that is being analyzed, and the genomic DNA used for the analysis is derived from the same source. So anyone who would like to answer this? Okay, option D. So yes, the correct answer is option number D. So the genomic DNA, which we are trying to analyze in the next generation sequencing, is heterogeneous in nature, is fragmented. Then we form multiple clusters of that, and then we perform the sequencing and analysis. The next question is a sequencing method where the visible light generated by the luciferin mediated reaction is collected as the raw data and the base sequences are identified. The type of sequencing method being referred to is it Sanger dideoxy method, Maxam Gilbert sequencing method, pyrosequencing, or dideoxy sequencing method? So option C, okay, right. So it is the pyrosequencing method. As we know, in pyrosequencing, the pyrophosphate which is released leads to the luciferin mediated reaction generating the light. The next question is, next generation sequencing involves sequence dependent DNA-DNA interaction. Which one of the following options is correct with regard to the mode of interaction? So is it the complementary sequence? Is it the hydrogen bonds held, or they are held together by the fluidic chamber, or there is some electrostatic force of attraction? Interaction. Okay. So yes, it is the hydrogen bonds which are involved. So these hydrogen bond interactions are involved in this sequence dependent DNA-DNA interactions. Next is, which one of the following objectives can be met with the next generation sequencing approach? Can we perform whole genome sequencing, or can we only perform target sequencing of exons, or RNA sequencing is possible, or whether all of these is possible? D. Okay. So, right, as I mentioned in the previous slides as well, that NGS is not only limited to this DNA sequencing, but it can be used for the sequencing of the whole genome, or we can target the exons also, which is known as exome sequencing, and RNA sequencing, as I mentioned, RNA-seq. So, this is a diagram, this is a schematic diagram or the depiction of the image for a DNA sequencing gel using the Sanger's method. Now, based on your understanding about this method, identify the option that shows the DNA sequence of the template strand which we have used. So you can take some time and you can think like what is the DNA sequence of the template. Okay, so Som has answered option number D. Anyone else who would like to try? Option A, Smriti. Okay, Vicky also answered option D. Does anyone like to explain why they have chosen this option? So the correct answer is actually option number B. Uh, Anyone who can tell why it is the correct answer? I think I need to explain this gel reading again. So in the Sanger method, we know that the smaller fragments will be obtained at the lower side and the fragments which are of larger size will be obtained at the upper side of the jet. So we start reading from this lower end. So firstly, we can see that first band which is obtained is in the lane number 3, which consists of DDCTP. So we know that in the firstly, what we need to understand is that when we are reading from this lower to higher side, we are reading from the 5' end to the 3' end of the complementary sequence. But what I am asking is, I am asking the DNA sequence of the template strand. So this will be, the first residue will be, according to this, it should be C. But this C is present at what side? This C is present in the 5' end of the DNA which is getting formed. Like, okay, wait. I think I should go to this slide. Okay. I think I should explain this slide again. So this will be better. Like we have a template strand in direction 3' to 5'. Our primer binds to the 3' end of the template and our extension or takes place in the 5' to 3' direction. So whatever the new DNA is forming, it is forming in the direction of 5' to 3'. So in this case, what happened is, uh, yeah. So in this case, this will be the 5' end of our newly synthesized DNA. But if I am talking about the template strand, then this is representing the 3' end of our template strand. Is it clear? Is it making sense? Is it clear? Yeah. The DNA sequence being generated is reverse complementary to the template strand of the DNA. Okay, right? So is it clear to all why it is option B? Okay, Ricky, you still have a doubt why it should not be D?
Yes, ma'am, you explained it.
Okay. So, see, this you have mentioned it correctly that the DNA sequence being generated is the reverse complementary to the template strand of DNA, right? You know this. So, but that will be in what direction? In 3' to 5' or 5' to 3'? So, the reverse strand, see, in this case, it will be the, okay. Okay, I think I should write it. So when we are doing, when we are reading this, so this is the 5' end, right? And this is the 3' end, right? Of the template, of not of the template, but of our readout, which is the newly synthesized DNA. This means for our template, this will be the 5' end, and this will be the 3' end. So whatever sequence you are getting from here by reading 5' to 3', hello, I am talking about the template strand. Reading should be in 5 to 3. So what is the doubt then? Is it clear, Ricky? Or is it clear to others?
Yes, ma'am. Because of the fact the template strand is itself is the complementary to the sense strand, which is 5 to 3, and the template strand in actual it is 3 to 5.
Yes,
But since the gel is generated 5 to 3, then we have to make it reverse complementary to match the
right,
sequence of the answer.
right. So, like you have understood the concept. Is it clear now? Okay,
Yes, ma'am.
okay. So the next question is, shown below is a schematic of an assay platform which is used for the genomic approaches. And so which one of the following option correctly identifies the property of the probe which are indicated here? This is a Fe matrix chip. So what is the property of the probes which are used here? Option number D. They are fluorescently labeled DNA. Okay. They are oligonucleotides. Okay. So yes, the correct answer is that they are oligonucleotides. This Fe matrix chip is basically used for the microarray. So in this, our probes are not fluorescently labeled, but we fluorescently label the desired DNA sequences. Okay, so in this case, the schematic here represents the gene chip, and the probes here are oligonucleotides that are complementary to the mRNA sequences or to the target DNA. However, in this case, the probes are not fluorescently tagged. Okay. The next question is, the schematic diagram shown below represents which one of the sequencing methods listed? First is Sanger dideoxy method. Second is Maxam Gilbert sequencing chemical method. Third is pyrosequencing, and fourth is none of these. This is very easy. So it is clearly pyrosequencing. So the schematic here represents the pyrosequencing method, where the visible light generated by the luciferin mediated reaction is collected as the raw data and the base sequences are analyzed. So I'll end today's discussion here. If anyone has any doubt, they can ask regarding any of the sequencing techniques.
I have actually.
Yes,
The shotgun sequencing, what are the differences between shotgun and the Sanger sequencing, which makes what makes it different? And does it have the requirements same as the requirements for Sanger sequencing, or to some extent it is different in terms of the ingredients it requires?
okay. Anyone else who has any doubt? Okay, I'm explaining this. So, so you asked what is the difference between the Sanger sequencing and a shotgun sequencing, right? So in shotgun sequencing, we need a library. So firstly, what we do is in the our original genome is broken up using various forces like we can use various forces like ultrasonication for the fragmentation or DNA molecules. leading to the generation of many small fragments, and then these small fragments are cloned, right? So the next step involves the cloning, which is not there in the Sanger sequencing. In Sanger sequencing, it is basically, see, Sanger sequencing is based on the replication principle. So it is basically terminating the replication of the DNA synthesis, right? While on the other hand, the shotgun sequencing is not based on the termination of the replication synthesis, but it involves the sequencing of many smaller fragments, and then it tries to identify the overlapping regions. So wherever there is a overlapping between the two different contigs which we have cloned, because whenever we are fragmenting, it is not like the fragmentation will be uniform. There will be some heterogeneous fragmentation. Therefore, millions of copies will be there, which like suppose there will be many copies, and these copies might have some overlapping hand. So this overlapping ends actually helps us in identifying, like if we are starting from A, B, C, D, E, F, G, H, and the other has E, F, G, H, I, J, K. So we know that there are certain overlapping sequences which are present in both of them. This means it might come after this. So this is how we sequence in the how we perform sequencing in the shotgun sequencing. While in case of Sanger sequencing, we use different ddNTPs which are fluorescently labeled, and based on these different ddNTPs which gets incorporated during the time of DNA synthesis, DNA strand synthesis, that leads to the termination of the chain, leading to the fluorescence, and which is recorded, and thereby we sequenced using that fluorescence signal. Is it clear?
But does it require the DNTP and the DNA polymerase like Sanger sequencing? If I ask about the shotgun, look like uh, the Sanger sequencing requires the DNTP and DNA polymers as well. So does it require does the shotgun sequencing requires the same as the Sanger? The process would be the different. Obviously, you explained it, but the ingredients is there any change or different from the previous one?
So it will be basically, just a second. So I guess you are asking that when we are cloning, after cloning, then when we are sequencing, on what basis we are checking the that overlaps. Am I right in saying this?
Yeah, in terms of ingredients, because of the fact that DNA polymers and DDNTVs are required for Sanger sequencing.
Yeah.
For the shotgun sequencing,
Mm.
it
So
does uh, the same. Maybe I don't know that the technique.
see, the technique is different, but yes, as you said, uh, we need the similar kind of like we need the similar thing here also when we have to sequence these smaller fragments. So in order to sequence these smaller fragments, in order to know the sequence of these smaller fragments, the sequencing here will also be similar to that of Sanger sequencing. The difference is only that here we are using the complete genome and then we are fragmenting it. So yes, in order to like I think you are saying that how we are then sequencing these contigs basically, right? After only knowing the sequence, we'll be able to find the overlapping sequences. So
Yeah.
this sequencing is based on that similar method, but the shotgun sequencing here just differs in the way that it leads to the sequencing of the larger fragments of the DNA, because by breaking up the DNA molecules into these shorter fragments.
Means it is different in terms of the technique it uses, but not in
Yes,
terms of
yes,
ingredients.
yeah,
Both are same
yeah,
in
yes,
terms of ingredients.
yes.
Okay.
Is it clear? Anyone has any other doubt?
One more confusing and like term I have seen, like the multiple sequence alignment. So what is its usage in our research, or how it can be used in an effective way, so that it would be useful tool for us? Like multiple sequence alignment, is it the alignment of the genome in a graphical manner or something else? I didn't get anywhere the term which is being
See,
taught.
okay. So this multiple sequence alignment, if I'll go to the alignment I'll explain in the next class again. So basically alignment means when you are trying to align the two sequences, whether they have a similarity or not. So suppose I have a sequence where I have, let's say I have a protein sequence where I have XYZ, something of this sort of sequence, and I have another protein sequence where I have YZA. So I'll try to align whether these two sequences have any percentage of similarity or not. So this is the alignment. So in case of multiple sequence alignment, what we do is we use the multiple sequences. Multiple sequences means multiple sequences from multiple organisms. So suppose, for example, if I want to know if there is any in case suppose if there is a protein which is functional protein, and if there is a but I don't know what is the functional unit of that protein, like what is the domain which is, where is the domain which is present, what can be the probable motif which is present. So what we can do is we can take multiple sequences from different organisms like of human, zebrafish, drosophila, of xyz organisms. You can take multiple sequences of those proteins and you can try to align them. Since we know that in nature tries to conserve those sequences which are important for the functioning of the protein. Therefore, the sequences which are required for the functioning of that protein, we expect them to be evolutionarily conserved in these species, across the different species. So, by aligning these different protein sequences with each other, or by performing the multiple sequence alignment analysis, you can get the information of that type. Does that answer your question?
So can we use this information for production or the manufacturing, or using this information, valuable information for developing a transgenic model for disease detection, like knockout?
So basically, this will actually help you in knowing the your this will basically help you in knowing what is the importance of what sequences. Suppose there is a sequence which is highly important. So this will provide you the information about that sequence, and if you want to see whether if deleting that sequence will have anything any effect on my phenotype or the genotype, so you can see that by deleting that sequence you can definitely check. So multiple sequence alignment is the basic step that we usually perform in order, is the bioinformatic analysis which we usually perform to see like what is that important fragment, or what, which is the important region. And by knowing that important region, you can then perform the gene knockout or gene knock-in, whatever you want, whatever your study is. So you can just knock out that segment, and then you can check whether there is any change in the function or not, which is taking place.
So this is how we start looking and comparing the sequences amongst
Yes,
organisms.
yes, yes, yes.
So is there any specific software is there, or there are multiple softwares available
There
across
are
the?
softwares available for the multiple sequence alignment.
Mhm.
So there are multiple softwares, like you can go and feed the sequences there, and then you can perform the multiple sequence alignment. So there are like EMBL, that is the European Molecular Biological Laboratory, has its own tool for the multiple sequence alignment, and similarly there is a tool which is the Clustal W. So there are many tools which are used for the alignment of these multiple sequences, and I'll discuss all these tools, or whatever which are the most commonly used tools, in the next class, if you want me to discuss that also. Like what is local alignment, what is the global alignment, what are the different
Yes,
tools which are used.
that would be really helpful in the next class if you can do so.
Sure, sure. I'll add all these things
Thank you.
and I'll discuss all these things in the next module.
Thanks a lot.
No problem. Any other doubt if you have, or
Uh,
anyone else, if anyone has any doubt?
so So one more doubt: if anyone couldn't ask any question, then I can move forward with one question I have for today, because it's already extended the session.
Uh,
It's already 2 minutes over.
okay. Okay, it's okay. You continue if anyone don't have. Anyone else has any doubt? We'll just have one more last class next week. So if you have any doubts regarding the previous modules also, you can ask in the next session as well. Or in today's session, if you have any doubt, you can just write it in the chat box or you can unmute or ask. Yeah, I think no one has any other doubt. So if you want to ask it today, Ricky, you can go.
One simple question that uh, before checking all these sequences, whether for the disease detection or for just for the purpose of research for being a student of PhD or postdoc, but what I need to ask that before we start looking at the sequence, what one thing we should do or we have to do or we are doing, that isolation of the DNA and RNA, that's really important. Do you know, means but there is less information available in this module or in this course how to isolate the DNA and RNA in an effective manner before we start proceeding to other techniques.
Okay, so you want to explain how to isolate the DNA and RNA.
Yes.
So it it is again a long procedure. So if you want, it will be better if I'll add those
Next
slides.
class would be fine.
Yeah, in
Next
next
time,
class
yes.
I will explain those. But okay, so you want me to explain how to isolate DNA and RNA, and then how to proceed with this multiple sequence alignment thing.
Yes, the
So
whole
okay, in the yeah, okay, so
synchronization of the techniques from
okay,
first to
okay,
last.
fine. In the next class we can have that only if all of you agree, as it will provide the idea also that where and when to apply what technique. So I'll make a brief schematic diagram representing where and when we have to apply what techniques, and I think this will help in summarizing this course overall, like overall summarizing this functional genomics course. So yeah, sure, Ricky. Thanks for the suggestion. I'll add this in the next class.
Thank you, ma'am.
Okay. So if no one has any other doubt, I will end the meeting. Thank you. Thank you for attending.